Yes, I've forgotten the most important thing. I'd like to welcome Andrea Morales Rosso.
Eh, Buenos días a todos. Voy a hablar en español, entonces los invito a que usen el código de traducción simultánea. El día de hoy les voy a presentar un trabajo que hemos realizado con una que fue estudiante mía de pregrado. Se llama Lady Gómez, pues ya se graduó, pero seguimos trabajando en el tema. Y con otra profesora de la Universidad de Los Llanos, que es Luz Estela Suárez, que es la directora del Herbario Llanos, y dos colegas de la Universidad de Antioquia y de la UPTC. Entonces, muchos de los servicios ecosistémicos que están en las ciudades son proporcionados por las plantas, como la regulación de la temperatura, la fijación de carbono y otros soportes pues, para la biodiversidad. Y las interacciones entre las aves frugívoras y las plantas pues, son fundamentales pues, para el mantenimiento de las mismas y se pueden estudiar a través de las redes de interacción. Entonces, aquí vemos un ejemplo de una ciudad en Curitiba, en el sureste de Brasil, donde vemos que a medida que aumenta la urbanización, hay una pérdida en la riqueza de aves y encontramos redes que tienen pocas interacciones, que tienen pocas especies, y pues la pérdida de ese hábitat se traduce en la pérdida de especies especialistas y vamos a encontrar entonces redes donde las especies generalistas son muy abundantes y se alimentan de los frutos disponibles e incluso de frutos de plantas exóticas. Entonces, ha tenido una hipótesis donde la urbanización actúa como un fuerte filtro para las relaciones que se encuentran en las interacciones entre estas especies y donde va a haber una prioridad en facilitar esas interacciones para las aves generalistas y va a haber menos especies especialistas, va a haber cambios en ese anidamiento de esas relaciones y en esos módulos que se van a formar. Entonces, para poder estudiar estas redes, ¿cierto? Estas redes de interacción, hay una serie de métricas. Está la métrica del anidamiento, la métrica de la modularidad y la métrica de la centralidad. Entonces, nosotros para la métrica de anidamiento esperamos que para sitios con zonas urbanas, pues vamos a encontrar redes que son más anidadas, donde vamos a encontrar especies de aves que se relacionan con más especies de plantas. Y en sitios donde va a haber mayor cobertura de bosque, vamos a encontrar redes menos anidadas, donde va a haber especies de aves que se alimentan con de ciertas especies de plantas. En el caso de modularidad, esta variable está asociada a los subconjuntos de interacciones que podemos encontrar, entonces a la cantidad de módulos que se van a generar. Entonces nosotros esperamos que en sitios urbanos haya menos modularidad y que en sitios de bosque pues va a haber más modularidad en el sentido de que va a haber especies que se van a alimentar específicamente de ciertas especies de plantas. En el caso de la centralidad, que es como la importancia que tienen las especies en esas redes de interacción, entonces nosotros esperamos encontrar que haya mayor centralidad en sitios urbanos, es decir, que va a haber pocas especies que se van a alimentar o van a estar relacionadas con varias especies de plantas y en las zonas de bosque vamos a encontrar menos centralidad porque esas relaciones van a ser pues más específicas. Entonces nosotros trabajamos en Villavicencio Meta, que está en el piedemonte de los llanos de Colombia. Esta ciudad pues tiene todavía mucho potencial para planificar y tratar de mejorar el tema pues de conservación de la biodiversidad, ciudad todavía muy verde. Y escogimos dos lugares. Uno es el Jardín Botánico, que es un remanente de bosque que tiene 46 hectáreas y eh, está entre 500 y 600 metros de elevación, está como en la parte, digamos, alta de la ciudad. Y el Campus Barcelona, que es donde yo trabajo, que es una zona rural heterogénea con 40 hectáreas y tiene más o menos, está a 400 metros de elevación, pues está como más hacia la planicie. Entonces, utilizamos 10 transectos, o sea, establecimos 10 transectos, 5 en cada lugar, ¿cierto? Y en esos transectos, entonces realizamos observaciones en la temporada seca y en la temporada de lluvias. En cada transecto, entonces, realizamos observaciones focales de las aves, de las interacciones, y también tomamos datos de riqueza y abundancia de las especies de aves. Y pues se colectó todo el material de plantas que fue identificado y procesado en el herbario Llanos, y pues todo ese material, digamos, se sistematizó y está en el GBI. También entonces en términos de caracteres funcionales para las aves usamos abundancia y para las plantas usamos, por ejemplo, el tipo de fruto y la abundancia de frutos. Calculamos por transectos, se realizaron también buffers por transecto y calculamos el porcentaje de urbanización y la distancia a vías principales. Y queríamos ver el efecto de estas variables sobre esas medidas de estructura de la red que ya les comenté. Entonces hicimos modelos lineales generalizados pues para ver qué relaciones encontramos. Entonces, a manera general, encontramos estas dos redes por sitio, donde para Campus Barcelona hay un 11% de urbanización y para Jardín Botánico 4% de urbanización. En general, encontramos que las redes son anidadas, modulares y están principalmente compuestas por aves pequeñas, abundantes eh, y que son principalmente generalistas. En el campus Barcelona, entonces encontramos 51 especies de aves y 32 de plantas y vemos que es una red bastante anidada y no tan modular, que era pues algo a lo que esperábamos encontrar. Y en el caso del jardín botánico encontramos 50 especies de aves y 20 de plantas y encontramos que es menos anidada y pues sí se ven los módulos que era algo de lo que esperábamos encontrar. Aquí vamos a ver entonces un ejemplo de uno de los transectos del campus Barcelona, donde vemos ese, digamos, ese anidamiento de la red y pues no se ven claramente los módulos y vemos que hay especies principalmente generalistas de aves, sola pues una especie migratoria, pero pues ella llega, está un tiempo y se va y come pues básicamente los frutos disponibles. Y tenemos este ejemplo de esta especie, esta red en bosque, donde vemos claramente los módulos y está compuesto por especies principalmente especialistas. No encontramos relaciones entre la urbanización y las métricas de las redes, pero sí encontramos una relación entre la distancia a vías y el alineamiento. Entonces eso puede estar afectando o tener un efecto en las especies y sus funciones. Y encontramos una relación significativa entre centralidad y la abundancia de especies de aves en sus transectos y la centralidad y la abundancia del tipo de fruto y la abundancia de ese fruto, que ese fruto es como un higo con unos picos. Entonces, pues las estructuras de las redes son anidadas y modulares y están compuestas principalmente por aves generalistas. Son especies de aves que son pequeñas, son abundantes y consumen plantas que son de rápido crecimiento eh, y son pioneras y tienen unos largos periodos de fructificación. Y la urbanización pues no mostró pues, un efecto significativo en las redes, pero la distancia a las vías sí, y eso puede tener un efecto en las especies y sus funciones. Pues la idea es recomendamos seguir monitoreando estos efectos en la urbanización en estos tipos de redes. Entonces, esto fue financiado por la Gobernación del Meta y la Universidad de los Llanos, y pues hubo un montón de personas que nos ayudaron y nos colaboraron Muchas gracias por su atención. No sé si tienen preguntas.
Well, good morning, everyone. My name is Daniel Rojas. I am a PhD candidate at the University of Kansas and the Biodiversity Institute of Kansas. I would just like to start by saying that thank you for sticking out to the last day of the conference. I'm really happy to have you all here and to share you about this really beautiful work. And also that all you have enjoyed your days here in Bogota. Weather is kind of crazy, but it's so today I'm going to talk to you about a very charismatic species of the Caribbean. It's a snail of this size. It's called the queen conch. And if you have the chance to go to any beach in the Caribbean, you probably have seen them. Like this beautiful pink conch, and I have pictures later, but it's like this size. Many people collect them, and it's one of the most important fishery resources in the entire region. Um, More than 20,000 families, fisher families, depend on this. And for the last 50 or 60 years, it has been the top 5 fishery resources in many places like the Bahamas, Jamaica, Honduras, and other places. So this is the geographic distribution of the species. It's a species that lives at like at most at 30 meters of depth and less. And larvae of this species, so this is a mollusk, so early stages it disperses through larvae. Larvae follow the ocean currents. And so as you can see on that figure on your right, these currents go to the north. They go towards the entire like coast of South America and Central America. And they do this little loop over Florida, between Florida and Cuba, and then go northward. These larvae follow these exact directions. So connectivity, it's very dependent on the currents. Unfortunately, this species, like because of its very huge economic importance, has faced overfishing for the last 40 years. There's also other factors affecting its survival in the Caribbean, including the reduction in population connectivity. So because of the overfishing, like there's like less adult populations that produce more larvae that can go on the sea and then just then reproduce to other regions or disperse to other regions. So this also it causes this Ollie effect, which is like decreased population size and decreased survival probabilities in certain regions. So what we did for this project, we integrated multiple biodiversity data to propose or to like model to the best possible accuracy the distribution of these species to in different scenarios. We use occurrence data from OBIS and GVIF. It goes through this type of data always goes through a very thorough cleaning process. We know that like all the data is not equally usable. You have to go to some standards to clean it, like especially thinning it, like specimens that have been collected and not just observations, because it's very difficult to trace them back in terms of information and other types of cleaning. And so we used environmental layers to do these models because we want to test, we wanted to model what would be the distribution of these species under different climate scenarios for the present and the future. We use the variables available in Oracle, which is a very new system, offers very high quality ocean variables, including temperature, salinity, seawater, velocity, direction, and others. These variables, we use them for the present and the future, as I mentioned, under different scenarios of climate change and for 4 different 20-year intervals. And so for doing this, we have to do, for modeling this distribution of the species, we have to come up with a niche model, a model based on suitability and environmental suitability. And for that, we need to have a hypothesis of what's the accessible area of the species. This is the accessible area that we hypothesized based on previous studies. There's like many different ways of doing this, but this is one of the most recent and more thorough ways to do until now. And it's a good hypothesis based on the literature. So with the clean occurrence data, the environmental components or environmental layers, plus the accessible area that we hypothesized, we produce ecological niche models. These models basically estimate the environmental suitability of a species and the environmental conditions under which species can maintain populations over time without the incidence of immigration and other effects. And based on that, we produce some environmental suitability estimations. Additionally, we considered high-resolution seagrass habitat data. So seagrass habitats are very crucially important for this species. These species depend nutritionally on these seagrasses. We know that seagrasses have been like a threatened habitat across the world. Fortunately, in the Caribbean, they're still very well preserved in many regions. That's great news, particularly in the Bahamas. The Bahamas is paradise of seagrasses. And also we also evaluated what would be our distribution estimations look like under the current configuration of marine protected areas that exist in the Caribbean today. This was taken from the Marine Protection Atlas. It's a widely open source, accessible database, high resolution database. And so with that, we produce these estimations. So what you see here in red is the areas that are currently suitable that are expected to be lost in the future. So we have three intervals here. And the area that's in like dark blue, it's area that's all remain unsuitable for the entire time. And the blue area, which is the most important one in this case, and the yellow one as well, are the areas which are predicted to remain suitable across time and decades, at least until 2018, based on our models. As you can see here, basically the distribution is predicted to move northward. Most of the areas or suitable areas currently in South America are expected to be not environmentally suitable in the future. There are different explanations for this. But what we predict is that many places where there are still populations, particularly in Colombia, Venezuela, Honduras, Costa Rica, and some parts of Mexico, are probably be less and less suitable for the species. And our results relative to the seagrass habitat data that we got shows something similar across the two types of models that we produced. One of them produced with Maxent, and the other one produced with ellipsoids, which are two models for developing niche models. Predict a very overall broad and well represented distribution across the seagrass habitats. Maybe Maxent shows like some seagrass beds at the north part, like in Florida and North Bahamas, are gonna be less suitable. But this is a work in progress. We need to keep investigating this because there are several ecological processes going on here that they're just not completely explainable just by environmental factors. And this is our predictions on the current distribution of marine protected areas. This is super interesting because we can basically see how environmentally suitable are protected areas today and will be in the future if they keep like they are. So as you can see, many protected areas in the Yucatan Peninsula and the Central America will probably not be very good for the species in the future, to be honest. And many places in the lower Antilles similarly, and North and South America. So this calls for maybe a way of, or just to start to rethinking our configuration of marine protected areas regarding these species in particular. And this also, it's a method that's applicable for any other species, let me say that. But it's just a way of saying how, or my estimating, have a rough estimate of how effectively would a marine protected area would be environmentally tough. So there are a couple of conclusions that are very important about this. So species distribution models based on ecological niche modeling, are a very powerful tool for estimating suitability in terms of environments and climatic factors. However, there are other processes that these models do not capture always. These species in particular, as I mentioned earlier, relies highly in connectivity. So the currents in the Caribbean and the Atlantic are highly influential on how these species can access other areas. So probably what we would need in the future is would be translocation of these species of some populations because the currents naturally would not be able to move them to the areas where it could be suitable. species distribution models are a great tool for this, but still more researches need to be done. And evaluating suitability using species distribution models using other tools as a complementary tool, it's one of the cases where you can have the most effective and accurate predictions. With that, I would like to thank you. And I would like to thank the KUNM lab at the University of Kansas for all the support and help with this project. I'm happy to answer any questions at the end. Thank you very much.
Okay. Hello, everybody. I'm Tommaso, a PhD candidate in the Integrated Remote Sensing Studio at the University of British Columbia. And today I want to bring a more remote sensing focused perspective on how we can go away and map and monitor landscape configuration effects on natural disturbances. So like you're all very familiar with, we're experiencing a series of events that are changing the way that forests behave, species composition come back and forth. So in Canada, for example, and across the world as well, we have strong anthropogenic impacts, for example, harvesting that occur across large areas and tend to homogenize the landscape. Climate change, of course, which changes the landscape functioning and compositions, and then natural disturbances. So for example, occurrences of large fires, which increase in size, the frequency, and severity. On top of that, we also know that the landscape structure and configuration tend to affect how disturbances occur. So for the landscape structure, we can think about it, a distribution of, for example, age across the landscape, or the distribution of tree sizes across. Versus configuration is more around the arrangement of particular parts of the landscape and how they are arranged in space. So to give a more concrete example, if we look on the right-hand side, we might see a simulated landscape with the homogeneous areas that might represent a particular property of interest. So for example, individual species or developmental stages. So when we think about configuration, really we think about it in two different ways. We can think about it how configuration might affect the occurrence of disturbances. For example, the distribution of host species for an infestation. Or we can think about how disturbances in turn may affect the occurrence of particular arrangements, for example, of conifer patches. And so in particular here, we're interested in the former thinking about it. So how the configuration might affect the occurrence or the dynamics of disturbances. So a little bit more into how the configuration actually works. So we take the exact same landscape, with the distribution of a particular property of interest. Let's suppose these are conifers at different stages of development. And then for every single one of these homogeneous areas, we might calculate property of interest, like the area of these patches, what they look like, and how they are arranged in space in relation to one another. So, too many. Okay. So historically, how we go away and measure this configuration. So across many countries, you might know that we have a forest resource inventory, which acquire aerial photography or aerial imagery across a particular area. And then this imagery are given to photo interpreters who go away and manually delineate homogeneous area on the landscape, which represent a property of interest. So you might understand now that if we start to having analysis at larger scales and over time, this can become a little bit subjective because interpreters change and interpreters have different ways of aggregating these stands. So if I were to have the same aerial photography and give it to two of you in the room, you might come up with different strategies. Some may have more clamped arrangements, some may have more dispersed arrangement of particular forest patches. And so we're very lucky that we have a continuous monitoring from space, from Landsat in particular. So it's the longest continuous satellite mission that allowed us to extract information from the landscape at very regular and frequent time intervals, biweekly to weekly information. And so we can leverage that availability of long time series to extract the property of interest and to go away and automatically segment these stands from these consistent observations over time. So in this case, we see a period of time where Landsat imagery have been acquired. These, I believe, are yearly steps. And if we can feed this information into an automatic segmentation algorithm, then we can extract some stand information for a particular property. So this is more time consistent, where it's not subjective from an interpreter. It's an algorithm that goes away and uses that information to extract configuration estimates. So I think I skipped. So overall, we decided to ask, we were interested in three main questions. First, can we actually go away and get the configuration estimates? And can we capture disturbance dynamics based on these configurations that we can extract from space? And then secondly, are there properties of this configuration that are more sensitive to disturbances than others? And if we were to take a time series or temporal approach, does the influence of this configuration on the disturbance change over time? So to do that, we focused on, we had a particular focus species here, spruce budworm, Coristonera fumeraria, which is a native defoliator of fir and spruce species, particularly balsam fir, black spruce, white spruce, if you're familiar. And it tends to generate the vast partial stand mortality. So we have an infestation that comes in today, and then the mortality that we observe might come up in 4 to 6 to 8 years, depending on the severity of that infestation. And then we know that this infestation then altered the species, the structure and the composition of species in the landscape. Ultimately, because this is a highly mobile organism, it's reasonable to think that the distribution or spatial arrangement of host species on the landscape might affect how the disturbance changes and moves across the landscape. So as a study area, we focused on Lac Saint-Jean on the right-hand side, which is in the southern part of Quebec. This is a bit of a hub site where we have its data rich primarily. But we see on the right-hand side again, a cumulative infestation map. So from the forest resource inventory, we're able to extract areas that are known to be affected by the infestation. And so larger numbers over time indicate that there have been more infestations occurring in that area. So there's primary infestations are occurring in the southern part of the study area versus the north is a little less affected. So in terms of the methodology, we took 13 years of landscape observations and annual time steps that you see on the left-hand side. And then for every single one of these 13 observations, we plugged these into automatic segmentation routine that gave us information about how these stands are arranged in space. And then for every single one of these individual stands, we were able to extract some spatial characteristics relating to the area, shape, and cohesion. So how close or far apart are these stands to one another? Particularly, we focused only on conifer species because these are the primary hosts for spruce budworm. For the second step of the methodology, then we took all this data-rich information for the configuration of the landscape at individual time steps, and we plugged that into two different random forest models. One that ran individual time steps, annual time steps, and a second one that it's more cognizant, so takes into consideration the whole times. So we were then able to extract the infestation likelihood or probability that an infestation might come back given a particular landscape configuration for a given. Then these are some other things that I don't think I have enough time, but you know that the infestation moves across the landscape. We've seen that from the forest resource inventory. And so we pretty much limited our sampling area to where the infestation occurred for a given year. And then, so these are some of the results that we obtained. We see that from in this case, 2013 to 2020, it's a bit more discretized to fit within the slide. We see that the infestation probability, so the probability of an infestation to come back, given a particular landscape configuration, tends to increase as the infestation actually progresses over time in the study area. So we have bumps of infestation where it's slightly more severe in 2017, tends to decrease in 2018 and back in 2018. And then for a more discretized, view, oh, there's no, for a more discretized view, we had, I can go back to the previous slide here. We were able then to track how this infestation progressed over time, and this has strong agreement with what the forest resource inventory told us. So infestation progresses from an observation point of view, so the FRI information, and we also observe this from space in a spatially explicit fashion. So for every pixel, we have a likelihood of infestation to come back. And so these are the three, I can skip these parts here. These are the three main indices that we found to be most sensitive at telling us how the configuration affects these disturbances. And these relate to the area of the patches and the variability in the patch area. So ultimately, our observation was that for a greater fragmentation of the conifer species, where these are intermixed with the deciduous species, we observed an increase in infestation likelihood. So this has been corroborated by other studies in the same area, and these mechanisms might relate to landscape saturation effects, where the infestation moves and finds small areas of conifer forest, which are like host species, and infestation tends to thrive. So to briefly conclude, we see that remote sensing is a valuable tool to give us to investigate disturbances in a spatially explicit fashion and across time. And the landscape configuration metrics help understand like large-scale patterns of how some of these disturbances occur. So including these metrics in some forest management strategies is a good thing to do because help us mitigate these negative impacts of infestations increasing as, for example, climate change. So this is all I have for my presentation. Thank you very much for listening.
I'm surprised we don't see more. Five minutes.
Yes. Hello, everyone. Actually, I'm going to give a lightning talk, 5 minutes. I hope I can finish 5 minutes. Okay, my talk is about connectivity between freshwater and marine ecosystems. Okay, geobon has several thematic forms. One of them is freshwater form, and another is marine form. I'm actually one of the hotel marine bomb. We actually, in this conference, we first decided to have a session after considering the biodiversity monitoring in transitional waters. And there's only one paper submitted. It is my talk. So it comes to this session. Anyway, there's actually in between freshwater and marine, there's estuaries and brackish water, which is the coastal area. It's really important ecosystem, offering a lot of variable ecosystem services. So we are focusing on our research site, especially my site, Hokkaido, eastern Hokkaido. There's a very pristine watershed remaining. It is actually called Akeshi and the Kanbei Shimaya. It is the second largest wetland in Japan, and actually about 40 kilometers away. So there's actually good forest and large wetland. This is a gray area and brackish water, and then agriculture. Here, there's a university to university collaboration, actually education program. Kyoto University has a forest, experimental forest station, and Hokkaido University has a marine station. And we are doing ecosystem connectivity education to our students every summer, and then taking long-term data of both river and brackish water and the marine. So the system is actually something like this. So there's a lot of interaction between ocean and coastal area and even to the land. So there's a meteorological factors like sea fog, precipitation in the forest, and river input from the existing forest or from the cow farming area affect the water quality of the brackish water. And then there's a lot of animals and plants in the brackish area, especially seagrass. Eelgrass is very prominent here and actually affecting a variety of different types of animals, including oyster, clam, which is very much important in this area. So we are taking a lot of variety of different types of long-term data. I don't have time to explain everything, but it is fish abundance in one river. It seems declining over 20 years. And this is a seagrass cover in fish question. We found they decline when there's a large rainfall or typhoon attacks, which actually drastically change the water quality. And we are taking a nutrient content changes and also water temperature and water color, phytoplankton abundance, which we haven't tried any qualitative analysis yet, but actually we think that these different EOV will be analyzed together to understand how the impact of land, water, the freshwater and marine ecosystems interact with each other. And so finally, with ongoing climate change, there will be a lot of future changes will be expected, not only natural environment by climate change, but we are also having a lot of different changes in human activities and these changes. Especially this area is now facing a rapid human population decline. We will have less usage of the both land and ocean, which may affect the ecosystem as well. Okay, thank you very much for listening.
Good morning, everyone. I'm very happy to be here to discuss if anyone who's interested. I'm going to talk today about the Hermandad Marine Reserve. And for those that are not very familiar with this, this is a relatively new protected area, and that it's located in the northeast part of the Galapagos Islands in Ecuador. And it extends to the east towards Cocos Island in Costa Rica. It adds 60,000 square kilometers of protection to our current Galapagos Marine Reserve that has been there for over 25 years. And it's an important area because the last few decades of research here demonstrate that there's a number of organisms that use this as a corridor to move between Costa Rica and Ecuador. So cetaceans, elasmobranch sharks and rays, turtles, and other commercially important fish use this area a lot. Just after the creation of this protected area, in 2023, a management plan was put in place with different objectives, including the protection of its biodiversity, its ecosystems, its ecosystem services, to promote scientific work, scientific research here, and also to put in place a long-term monitoring plan. Now, to put in place this monitoring plan, 18 stations were defined in the Hermandad Marine Reserve, 15 within its boundaries, and 3 seamounts outside its boundaries. And what is interesting about this protected area is that contrary to the Galapagos Marine Reserve, this is a very pelagic part of the ocean, the shallowest point being around 800 meters and the deepest points over 2,000 meters. So you can imagine how challenging it becomes for an area like this to monitor. And if we think that this putting a marine protected area here has a purpose, right? It's protecting these migratory species, it's protecting these ecosystem services. The first thing that we need to do is understand how it looks, the biodiversity here, how it's changing through time. And to do this in a pelagic area that is hundreds of kilometers away from the closest port, it's a big job. So different institutions, national, international, private, and governmental, have come together to do this. And this monitoring plan is actually very comprehensive. It includes oceanographic cruises to gather information, physical, chemical information, environmental data. It includes visual observations and surveys. It includes remote sensing data, and it includes eDNA. And today I'm going to focus on a tiny bit of this long-term monitoring plan, which is BRAVS and eDNA. BRAVS, it stands for Baited Remote Underwater Video System. So basically, putting these frames with some canisters that contain bait and a few GoPros and deploying these in the water column. So everything that gets attracted to the bait can be captured by the cameras. And at the same point, in the same stations, collecting water for doing some eDNA surveys. Now, not surprisingly, the results that we get from both methodologies differ a little bit, right? Because each methodology has its own biases. For example, the BROPs attract mainly the things that follow the bait, while eDNA does not have this restriction. We also know that eDNA, it's a very sensitive tool that captures a lot more than BROPs alone, but we also have some things that are detected by both. After some stringent analysis and quality check, we detected nearly 100 OTUs, and close to 80% of these 100 OTUs were identified to the species level, which is good. But I have to say, we are focusing on big stuff, on vertebrate animal, and on vertebrate taxa. But it's important to mention this because for us, while eDNA has become a great tool to monitor, right, many areas, still a combination of methods is required if we want to have a more comprehensive view. The other thing that we identify is this bioregionality. So here you see in green the stations that are on the western part of the reserve, and on pink the ones that are on the east side. And the community composition differs between the two. And this is detected by both methods. And additionally, we see some extra differentiation on these 3 points, which are the seamounts outside of the marine reserve. This makes sense because these are the shallowest areas and do represent an important habitat for a lot of species, including cetaceans and elasmobranch. Now, when looking at the data in terms of conservation status of the taxa that we are detecting, and this is according to the IUCN categories, we see that seamounts especially are quite an important area for threatened species. And this, as I said, it's because we have a lot of cetaceans, elasmobranch, and big fish here. When looking at the same data in terms of human use, and this is species that are commercially important or that are important for other activities such as tourism, we see that in general, the Hermandad Marine Reserve does represent an important habitat, important area for species that we depend on somehow. Now, as I said just a few minutes ago, understanding is the first step to know if we're making good decisions or not, right? This was the very first baseline that we built through a big effort, big collaborative effort in 2024, and has shed light into some important things like the need to have monitoring efforts more continuously. We do see with the environmental data that there is seasonality in this area. So it's important to do this at least twice a year. But again, resources to work in an area like this can be challenging. But this first snapshot was actually really important for us to know how to move forward. Second, the relevance of both marine reserves that we have, the Galapagos Marine Reserve and the Hermandad Marine Reserve, is very high, right? Not only if we think on species that are important for conservation, but if we think also in species that are important commercially. This biogeographic that we are seeing, we are now testing two hypotheses. One is environmental conditions as a first thing. Of course, the westernmost part of the protected area has cooler waters, it's more productive, so it makes sense that the community composition is different. But it's also like this western side of the Hermandad Marine Reserve are also closer to an area that's been protected for over 25 years. So it's not crazy to think that there's a spillover effect there that it's influencing what we are seeing in nearby locations. So this is a long-term plan, and we are producing a lot of data, not only molecular, not only through the cameras, but also with other methodologies that were not presented here. And it's exciting for me to be here because what we have been wondering is what's next for us. How do we make this data accessible? How do we make this data fair, as we have been listening in every single session? So it's exciting for us to be here and to talk to people from OBIS and from GVIP and from anyone else who's interested on these monitorings that we're doing and how to put this in a larger regional context. So thank you very much. Again, takes a village. So there's hundreds of people behind this work that I couldn't mention in just 1 minute that I have left. So thank you, everyone.
Hello, everyone. I'm Eugene, and I'm from the University of Hong Kong. So today, I'm happy to have everyone here to come to my talk. I'll be presenting the best settings for extrapolating species distribution models. So species distribution, as we all know right now, is very important. It's important for protecting area, for protected area planning and management. It's also important for assigning conservation priority. For example, IUCN uses the species ranges to define their red list categories. And species distribution models, SDMs, is one of the main sources of species distribution knowledge for us. So species distribution models, SDMs, correlate species occurrences with environmental variables to construct a correlated relationship between habitat suitability and the environmental variables you include in the model. And this relationship can be further projected in any given environment to estimate the species distribution. Ideally, training data should cover all suitable habitats or environments for the species that we model. But it is not always the case. In reality, species distribution model training data does not always reflect the full range of suitable environments. This can be caused by habitat loss, for example, by eliminating suitable environments, or biogeographic barriers that prevents access to suitable environments. In addition, no analog climates in the future means that current and past distribution data does not always cover the future environment, future climatic environment that species will experience. So species distribution models will sometimes extrapolate beyond, outside of the range of environmental variables that environmental conditions that's covered by our data. And this is especially true when we're projecting across space, for example, when mapping invasion risk, or across time, for example, when evaluating climate change consequences. So extrapolation is basically projecting outside of the range of the environment covered by your training data. And extrapolation is intrinsically bad because you have no data for model calibration or model validation. And the more you extrapolate outside of the range, so outside the range, the more, okay, yeah. so the more you extrapolate outside the range of your, outside the training data, the more you extrapolate, and the more uncertainties you get in your model output. So now we know that extrapolation sometimes is inevitable for when we do species distribution modeling. How do we maximize SDM performance? Can we use some algorithms that extrapolate better, or model ensemble methods that maybe summarize the models, single models better? Or do we include potentially important environmental variables while risking model overfitting? Because we do not have perfect ecological knowledge at all times. So to test SDM extrapolation performance under different settings, we will need first species with known distribution for quantifying SDM performance. And for that, we opted for the virtual species approach. And second, we need methods to precisely control the extent of SDM extrapolation. And we utilize the latitudinal gradient of climate in this for this particular purpose. So we designed a virtual extrapolation experiment. And I first took 700,000 tropical Asian butterfly occurrence records from this data paper that I was also involved in, and then generated distribution of virtual species using SDMs. So we obtained around 1,000 virtual species. And we sampled occurrence records and pseudo absences from truncated latitudinal ranges, like this. So we exclude the southernmost data. And to test various SDM settings, we constructed SDMs using different SDM algorithms, different ensemble methods, and we removed we removed relevant variables for some of the models, and we added in noise variables for some of the models. We then projected each ensembled SDM to the full study area, including the areas where we have no training data sampled, so that these SDMs extrapolate. And we compared the projected distribution with the virtual distribution to calculate the SDM performance. Sorry. Okay, so come to the results. The x-axis is the different SDM algorithms used, and the y-axis is the number of false predictions. So the shorter the bars, the better the performance of the particular model. And the false positives and false negatives are represented in different colors. So first, as you see here, GBM, random forest, XGBoost tend to perform well in terms of false positives, while the other models, the other algorithms tend to perform well in terms of false negatives. And we can see that there is no single algorithm that performs best in terms of both false positives and false negatives. But ensemble model, including all algorithms, perform better than any ensemble of a single algorithm. In addition, when we only include the top 4 best performing algorithms, we yielded ensemble models with even better performance. In terms of ensemble method performance, we found that there is no best ensemble method, as in the SDM algorithm. Instead, ensemble methods is actually an additional source of uncertainty, because different ensemble methods are amplifying the results of different algorithms. For example, the mean method is amplifying the results of these algorithms, and the median method is amplifying the results of these algorithms. And as for SDM variable selection, we found that the removal of only 2 environmental variables resulted in significant decrease in extrapolation performance, while the inclusion of 4 irrelevant noise variables did not significantly impact SDM performance. So we can conclude that modern SDMs are robust against noise variables, even when extrapolating. And the cost of false inclusion of irrelevant variables is less than the cost of false exclusion of relevant variables. So as a summary, we found no all-rounder SDM algorithm, and the choice of ensemble method is a source of uncertainty that needs to be quantified and reported. Yes, in addition, we as modelers need to tailor our models to our needs. Because, for example, if you're modeling invasion risk, then false negatives will cost you more than false positives. And we need to use multiple algorithms which have good extrapolation performance. So if you want to have minimal false positives, you can use GBM, MARS, and random forest, which performs well in this perspective. Or if you want to minimize the false negatives, you can use CNN, CTA, and maxent. And we need to include and we can include as much possibly relevant as environmental variables as we need, because SDMs are pretty robust against noise variables. These are the references, and a big thank you for coming here. Thanks to my funding, and thanks to my supervisors, Alice and Hughes. Alice is right here, and I'm happy to take any questions.
Ensembles, despite being one of the most widely used methodologies recently in species distribution modeling, as you show, are often risky because uncertain. So what is your opinion on that, and what is the possible solution to report in a paper, and not only to take that ensembles, because they are very risky in some situations?
So thank you for questions. Very good one. I think like ensemble methods are actually a way to quantify the uncertainties between single models, right? So I think in that sense, it is doing at least some work pretty well. But like different ensemble methods have like they all also have the uncertainties too. So if we can like use multiple ensemble methods and like display the differences between them, like the range of the range between like the outputs of different ensemble methods, I think that can properly address uncertainty even between ensemble methods.
Okay, well, thank you all for being here. My name's Noah, and yeah, I'm going to talk to you about a really interesting method we've been developing that allows you to scale SDM predictions from pixels to parks while correcting for sampling bias. So we just got a super great introduction to SDM, so I'll just breeze through this. We know we need to monitor biodiversity and anticipate how it's going to change with the environment, and SDMs are great for doing this. They give us maps of ranges that can feed into very policy relevant indicators, such as those in the global biodiversity framework. And so now more than ever, we need to make sure that our SDM predictions are credible and robust. But unfortunately, there's a number of challenges to doing so. I'm going to talk to you about 3 of those challenges. The first one is what we call relative predictions. So because SDMs are typically fit using just occurrence data, we need to add in pseudo absences for the models to compare against. And if we add in just a few pseudo absences, then the model thinks that this species occurs in 70% of the grid cells. If we instead add more pseudo absences, the model thinks that the species occurs in only 20% of the grid cells. So these SDM predictions are not real probabilities, they're only relative probabilities. The second issue I'm going to talk about is sampling bias. So because the occurrence data is typically found near cities, such as Montreal, where I live, and Ottawa, where I grew up, if we use these models to try to predict, say, the range of a black bear, it says that the black bear lives near the cities, and it completely misses this huge unsampled area of the true range. So this is another big challenge. The third challenge I'm going to talk about is scaling. So as ecologists, we always talk about how patterns can look very different depending on the scale that you look at them. And so for SDMs, this could mean that if we have predictions at a little like 1 kilometer by 1 kilometer pixel level, we might want to ask instead, what's the probability of occurring in a larger area, such as like a national park or something like that. And this is a really interesting problem that I'm going to talk a bit more about. First of all, if we were able to take these pixel level predictions and park level predictions, it would allow us to, first of all, use data at both of those scales. So for example, we could use expert-based park species checklists in conjunction with point data within one model. And then we could also then make predictions at both the pixel level and the park level, which could be super useful for lots of area-based conservation initiatives. But this is quite challenging, and the reason is species tend to cluster. So let me explain what I mean by that. Suppose we have a species that occurs in 50% of the grid cells in a park, and completely randomly, each grid cell is independent, and we want to know what's the probability that it occurs in this little 2x2 park. this is basic probability calculus. We can calculate that it's a 94% chance that it occurs here. But suppose instead that, again, it occurs in 50% of the pixels, but it clusters together. Now, we see that the park often occurs in these big empty areas. And so the probability that it occurs in the park is definitely less than 94%, but it's hard to know exactly what that is actually going to be. So with these three challenges in mind, our objectives are to introduce and test a model which will incorporate data at both the pixel level and the park level, and then use that to quantify and correct for the effect of these three different challenges that I talked about, and as a result of that, get more robust SDM predictions. To not keep you in suspense, this is the model we are presenting. Basically, don't worry about the details of it, but the basic idea is we have a biased SDM. We can correct for the effect of arbitrary number of pseudo absences using this orange parameter. We can correct for the effect of having sampling bias using this red parameter. And that gives us a corrected SDM. And then we can translate that into park level predictions while correcting for the effect of clustering using this green parameter. Okay, so I'm going to try to convince you that this model is useful, works well in a couple different ways. Well, okay, basically to test this model, we simulated species distributions with some extra clustering baked into those distributions. From there, we simulated occurrences and then inferred the presence and absence within a network of parks. Then we sampled the pixel level occurrences in a biased way, more towards the south. And then we use this bias data to fit a biased SDM. Now, the biased SDM and the park data feed into our model, which then spits out a corrected SDM as well as park level predictions. And to fit the model, you just play around with these three parameters until the park predictions reflect the true park data as best as possible. So yeah, let's look at, I want to convince you that this works in a couple different ways. First of all, how do those three parameters change when you change the settings of the simulation? So if we increase the number of pseudo absences in the simulation, you see that this orange parameter responds consistently to correct for that effect. The other two don't respond. If we increase the amount of sampling bias in our simulation, we see that this red parameter responds to that to correct for it. And if we increase the amount of clustering in our simulation, we see that this green parameter responds to that to correct for that. So the model is able to distinguish between these three different challenges, partition those into these different parameters, and correct for those effects. But you might ask yourself, does that mean we actually get better predictions in the end? The short answer is yes, at least in this theoretical context. So we can compare our park corrected method to our regular bias SDM that we started with, as well as a standard method of bias correction called target background. And we can first of all ask, how well do the different methods predict the overall prevalence of? We see that our method has very low error on the prevalence, while the other two methods do not capture the prevalence very well. Because these ones don't have access to that presence absence park data, they're not really able to. Secondly, we can ask how well do they capture the, overall pattern of the true distribution using a distribution overlap metric. You see that our method does a little bit better than the other two methods, it's a small but significant improvement. And then finally, we can look at how well does each method predict the occurrences within the parks, the deviance explained of those presences and absences. You see that our method does better than the other two because the other two do not consider this scaling up with clustering challenge. To wrap it up, we have seen that this method allows us to do a number of things that we couldn't do super well before. First of all, we can utilize checklist data even when it occurs at larger scales, or regions or parks. It's really useful because that is sometimes what's available in remote areas that don't have other data. And by the way, myself and my supervisor, Laura Pollack, are involved in an effort to, I guess, compile a database of these kind of regional and park-based checklists. So if you think you can help us out with that, please come talk to me. Additionally, once we have this model, we can make predictions at these larger areas, despite the fact that you cluster together. And once we have this, we can also measure and correct for issues like sampling bias and few absences. And finally, we also get to estimate how much species are clustering, which is super cool because this relates to other concepts in macroecology like beta diversity and species area curve. Yeah, these are just some of the directions that this model and iterations of it could go in the future. And I'm super excited I got to share it with you guys today. Thank you very much for listening. Yeah, I'll be here.
My research investigating the link between phylogenetic diversity and the dilution effect through the introduction of a new invasive mosquito, Culex pipiens. To start off, I will talk about the dilution effect. This is a hypothesis that an increase in biodiversity will reduce the number or the prevalence of pathogens in a community. So on this left side, we see a forest with lots of biodiversity. This forest supports lots of different animals. We have this mouse that is our competent host of Lyme disease. And because it's present with all these other species, our Lyme disease prevalence is lower, or our Lyme is lower. But if we take this forest and we plow it and put in a monoculture, it no longer supports this diverse community of animals and only supports this mouse, which is our competent host for Lyme disease, and our Lyme disease prevalence increases. However, when we look at actual literature, this relationship is not always clear. There's difficulties determining how to measure biodiversity, what factors to include, and what that shape of the relationship should look like. And so on this figure here, we have on the y-axis the prevalence of pathogens per host, and then x-axis our biodiversity. We have two different shapes for dilution effect here, our left skewed and right skewed bell curves. Is showing the difference in how much biodiversity it takes before we see a dilution effect. So our left curve, it happens pretty quickly, but on our right curve, it takes a while for us to see a dilution effect. And so there can be difficulties determining if we have enough data to see a dilution effect, if we have enough biodiversity. And then in the middle, there's the acetope, and this is showing the amplification effect. This is where an increase in biodiversity actually increases our pathogen prevalence. And this could happen, for example, if we have an increase in competent vectors or animals that are able to transmit diseases. And recent work has started to untangle these complexities. And one of these possible ways of capturing dilution effect is phylogenetic diversity. This is a means of capturing the shared ancestry of a community, and it takes a calculation by summing up all the branch lengths of a phylogenetic tree of the community. So on the left side, you can see some phylogenetic trees. When we have all the branches included, our phylogenetic diversity is 24. But as we cut away our branches, we're creating a community that is more closely related to each other and has less distance between each other evolutionarily, so our phylogenetic diversity decreases. And this may capture our feature or functional diversity better than traditional measures of diversity. And this relationship has yet to be investigated in vector-borne pathogens. Which brings me to my study system. This is Culex pipiens, a mosquito that was recently discovered in my province in Canada that you can see starred on the left. This is a province called Alberta that is in the prairies. This mosquito is important because it's a vector of many important pathogens such as West Nile virus, avian malaria, St. Louis encephalitis virus, and more. On the right side, you can see my collection site distribution. I also have my sites divided up into natural and urban sites. And for my methods, I aim to investigate mosquito diversity and pathogen prevalence across these different habitats. I use two different types of traps, one that collects mosquitoes that are looking to lay their eggs by attracting them with hay-baited water, and the other one is baited with CO2 or dry ice to attract mosquitoes that are looking to feed on a host. And then we also use a backpack aspirator that collects mosquitoes that are resting, and this was done on a weekly basis. And briefly going over the diversity that I found. Out of about 40 known mosquito species in Alberta, there was 21 mosquito species that I collected. 2 of these were Culex species, one of them including my Culex pipiens invasive mosquito. 16 Aedes species. This is as expected because Aedes is the most diverse genus in Alberta, and this was over 5 genera that I collected. So of note, only Culex pipiens was an invasive mosquito collected. All the rest of the species collected are considered native to Alberta. And so here's the raw diversity across sites. Again, this is colored by natural and urban. Natural is green, blue is urban, and overall our natural sites have a higher number of species, and our urban have lower, except for one site of urban has a higher level of number of species. My analysis for mosquito diversity will include 2 routes. One, looking at traditional measures using the vegan R package to calculate Shannon's index and Simpson index. And also looking at phylogenetic diversity, I will take an existing tree that is in the literature. I will use the APE R package to take that phylogenetic tree and make it into matching the community that I found at each site. And then from there, I'll use the PCANT R package to calculate phylogenetic diversity and species richness. And looking briefly at our phylogenetic diversity, a preliminary calculation, you can remember that our previous raw number of diversity, we saw that our urban sites had lower number of species than our natural sites. This continues to be true, but our one site that had a higher number of species for urban is now lower again. And if you look at the 2 natural sites, WCC and IBS, before they had a very similar number of mosquito species, but when we look at phylogenetic diversity, they're quite different. And this is showing the importance of looking at the shared ancestry of these 2 sites and how different they are. And I have some preliminary pathogen detection data as well. I was looking at avian malaria and West Nile virus. So first looking at Plasmodium, this is the genus of avian malaria that is transmitted by mosquitoes. Looking at all my Culex pipiens pools, they were all negative. And when I looked at West Nile virus, I did 26 pools, including all my Culex species again. And 10 of those pools came back positive, including 7 of my invasive Culex pipiens and 3 of my native Culex tarsalis. So this data suggests that Culex pipiens is an important vector of West Nile virus in Alberta, but not of avian malaria, but more research will be needed to confirm this pattern. To summarize, I found higher species richness and phylogenetic diversity of mosquito species and natural sites in Calgary, Alberta. I found preliminary detection of West Nile virus, and Culex pipiens, but not avian malaria. I will continue pathogen detection, and once I have this data completed, I can do my statistical analysis of pathogen prevalence with diversity to confirm if there is a dilution effect found or not. And these are my references. And thank you so much for coming to my talk.
My talk will be in Spanish, but Buenas tardes, yo soy Ángela Cuervo y vengo del Instituto de Biología de la UNAM y hoy voy a hablarles un poco sobre los vacíos en el conocimiento espacial de las plantas vasculares de Mesoamérica. Okay. My slides are in English, so I think that might help a little bit. Well, las preguntas que nos hicimos para medir los vacíos en el conocimiento fueron básicamente 3. Queríamos saber qué tan completo está el conocimiento espacial de las plantas vasculares, qué tan antiguo es ese conocimiento que está depositado en las colecciones científicas y si estas plantas vasculares están representadas filogenéticamente. Adicionalmente, evaluamos cómo estos vacíos del conocimiento están siendo amenazados por el cambio de uso del suelo y por el cambio climático. Para esto, utilizamos la información de GBIF, descargamos toda la información de plantas vasculares para América y eso corresponde alrededor de 18 millones de registros en el 2022. Pero como nos interesaba tener información a nivel de especies, solamente nos quedamos con 16 millones de registros a nivel de América. Este estudio tuvo un enfoque hacia Mesoamérica, que corresponde de los países que van desde México hasta Panamá. Entonces, al solamente enfocarnos en esos países, pues para la región existen reportadas alrededor de 32,000 especies aceptadas y eso redujo nuestra muestra de trabajo a unos 3 millones de registros. Este trabajo hace parte de un número especial de New Phytologies en el donde se invitaba a utilizar los catálogos de Q. Por lo tanto, toda esta información fue homogenizada hacia los catálogos de Q que se liberaron en el 2022. Y esto nos lleva a saber que para la región existen, Q reporta alrededor de 34,000 especies, 35,000 especies de plantas vasculares nativas. Y como van a ver más adelante, en términos de representación de lo que hay en los catálogos, como lo que está en GBIF, existe un buen porcentaje de especies reportadas en catálogos y en GBIF. Y hicimos análisis a 3 diferentes tamaños de celda, 60, 30 y 15 minutos de resolución. Yo les voy a presentar hoy la resolución de 15 minutos, que corresponde más o menos a 27 kilómetros. Bueno, lo primerito que hicimos fue hacer una homogenización con los catálogos de Q. Entonces, básicamente es renombrar las especies que están en GBIF con los catálogos de Q. Y para eso lo que encontramos es que existe a lo largo de toda la región una alta representación, más o menos el 90% de lo que hay en Q está representado en GBIF, aunque para algunos países como Honduras la representación es un poco más baja, va al 78%. Luego hicimos una limpieza geográfica de los registros, eliminando coordenadas que estén en 00, centroides, que estén cercanas a instituciones. Bueno, para eso utilizamos esta paquetería para hacer la limpieza geográfica. Y también hicimos una limpieza para saber cuáles eran especies introducidas o especies nativas según los mismos polígonos que propone los catálogos de Q. Con nuestra información limpia nos dimos a la tarea de responder nuestras 3 preguntas, donde la completitud la evaluamos con el paquete de NoVR, que básicamente mide para cada una de las gadillas qué tan completo está este inventario, qué tanto ha sido el esfuerzo de muestreo a lo largo de los años y que también puede ser considerado como un inventario completo. Posteriormente nos dimos a la tarea de medir qué tan antiguo, qué tan vigente está el conocimiento en esas celdas, en cada una de las celdas. Y básicamente lo que hicimos acá fue, de todas las especies disponibles, agarramos el año más reciente de colecta y calculamos una mediana de la data de ese inventario. Por último, en el aspecto filogenético, lo que hicimos fue saber si las especies que estaban en cada una de las celdas tenían secuencias en el gene bank, tanto de nucleotídicas o de plasticídicas, perdón. y también se estaban colocadas en dos grandes árboles de plantas vasculares, uno de Brownies beet y el otro específicamente de helechos. Entonces, con esto básicamente lo que hicimos fue ver si hay secuencias para las especies y si están ubicadas en los árboles. Posteriormente hicimos un tipo como de análisis espacial para agregar las celdas y poder combinar nuestras tres mapas de brechas de vacío y lo relacionamos con disturbios antrópicos. Como resultado, obtuvimos que en general pues estos índices disminuyen conforme se amplía la celda, por eso nos estamos enfocando en la de 15 minutos. Lo que vemos en términos de la riqueza documentada es que existe una alta concentración de registros en Costa Rica y es donde mayor cantidad de especies documentadas hay, pero está básicamente muy relacionado con la cantidad de registros. Estas gráficas que van a ver, entre más oscuro el color, significa que el vacío es mayor. En este caso, la riqueza es mayor. Y también tenemos zonas a lo largo en el centro del país donde la riqueza documentada es mayor, pero es donde también se encuentran uno de los principales herbarios, por ejemplo, de México. Vemos una pérdida de riqueza o de riqueza documentada hacia los límites, ¿no? Hacia el norte del país y hacia Panamá. En relación a la completitud del inventario, ustedes lo primero que van a notar es que existen vacíos, celdas en blanco, es básicamente para toda esta región, solamente fue posible, aunque tuviéramos información, solamente fue posible calcular la completitud para el 65% de las celdas. De estas celdas, todo lo que está en colores oscuros son valores de muy baja completitud, son celdas que tienen muy baja completitud. Y lo que resalta es, por ejemplo, Costa Rica, que tiene alta riqueza documentada, tiene también una baja, es más o menos completo, ¿no? Lo mismo lo vemos hacia Baja California. Entonces, aquí estamos empezando a ver una correlación entre el número de registros que tenemos, que podrían hablarnos de riqueza, y también con la completitud. Entonces, no siempre tener altos números significa pues, una buena completitud en los inventarios. En relación a la edad de estos inventarios, lo que vemos es que a lo largo de la región, la mayoría de las celdas, las especies no han sido documentadas hace más de 40 años. O sea, tenemos información, estamos trabajando con información de especies que no ha sido documentado a partir de los 80. Nuestros modelos de nicho, todo lo que estamos haciendo de análisis, ya no sabemos si realmente están ahí. Estamos trabajando con fantasmas. Okay, sí, más o menos eso. En términos de, también vimos esta edad a lo largo de la filogenia, ubicamos la edad a lo largo de la filogenia y vemos que a nivel de familia, muchas de estas especies tienen también más de, o sea, fueron colectadas hace más de 20 años. Y en relación a la diversidad filogenética, lo que vemos es que, bueno, hay una alta representación en las secuencias del gene bank y una que es del, perdón, en árboles filogenéticos está más o menos la mitad de las especies representadas y en el gene bank está más o menos el 70%. Los vacíos se dan principalmente hacia el norte del país. Costa Rica sí, a pesar de que era antes un país con buen esfuerzo en términos de la parte filogenética, es bajito. Y esta es la parte en la que combinamos estos vacíos en el conocimiento con los disturbios antrópicos y lo dividimos en dos zonas, en un hotspot y en un cold spot. Hotspot son zonas donde hay altos vacíos en el conocimiento en estas tres brechas y cold spot lo contrario. Básicamente lo que vemos es que el aspecto temporal está fuertemente ligado con la pérdida de cobertura, o sea, estamos en zonas en que tenemos conocimiento viejo y además tiene un disturbio antrópico importante. Y en la parte de los cold spots, lo que estamos identificando es que a pesar de que podemos entender que tiene información relativamente completa, en las condiciones climáticas actuales son muy diferentes a lo que a cuando fueron colectadas inicialmente. Ahí puede haber una presión en términos de climas no análogos. Y eso es todo. That's all. Okay, so next speaker Thank you. For everyone. So, so. ¿Sí? Listo. ¿Ya hacia dónde? Ah, okay. So, good afternoon. Thank you for coming. I'm Sara Colmenares. I'm coming from the Environmental Authority of Bogota, a biologist and a PhD in ecologist. I'm the data data analyst of the Monitoring Biodiversity Group, and today I'm here presenting is Juliana with me, our team leader, and our deputy Ivan Mel. This is one of the main results of the program of monitoring biodiversity in Bogota, who was created in 2021. And we made an effort on making protocols to measure correctly the biodiversity of the city, especially in the urban region, because Bogota has 25% urban area and 75% is rural. So this is just for the urban matrix. So just remember, the Singapore index was proposed and one of the conventions of biological diversity around 28 and 2010 integrates information of biodiversity, mainly birds and arthropods, for vegetation coverage, ecosystem services, and governance. And this is a self-assessment on how the management of conservation is going on in a city. So this index allows to compare cities not as a competition, but as a way to share information and to learn how to make it better. To make it's also able to make conservation efforts visible. It makes a strong point on assess correctly the biodiversity in an urban environment, identifies how human activities affects urban ecosystems, and all these topics are allows to enable planning and execution of policies that can balance the urban development along with environmental conservation. So it is composed by two parts. The first one is the profile of the city when we characterize from the geographical, socioeconomical to the political features. And the second part is where the data are important, where are 28 indicators distributed in three main topics: the native biodiversity in the city, ecosystem services, and governance management of biodiversity. So this is not the first time this index is calculated in Latin America. We have this calculated in 2023. Next year is going to be the update of this index for Bogota. The past year, Cali launched its results at the top. Well, it's above us. And other cities and before calculated Curitiba and Sao Paulo in Brazil and Mexico in North America. So just to put regionally, we occupy the third place among these cities. So the results, the profile of the city, this is a big savanna surrounded by a cordillera, the Eastern Andes, 2,600 meters above sea level. We have plains in the northwest, eastern hills, and the Sumapaz Mountains. We have a mean temperature of 13 degrees, around 800 millimeters of rain. We have two seasons, wet season and dry season, and the intermediate seasons. And we have a huge hydric system composed mainly by 5 rivers: Bogotá, Fucha, Tunjuelo, Salitre, and Torta.
And also we have this area, urban area, and a population around 8 million people. And one is important for the city is that we have 9 ecosystems. All of them are principally corresponding to the Andean forest types. Then the paramos and subparamos, the xerophytic shrublands in the south of the city, the floodplain forest, the wetlands, and the rivers and streams. One important thing is to, in the Andean and the wetlands, was declared the system of protected areas, which is composed, you can see them in the map. It's composed by sustainable landscapes, where to promote sustainable uses of and restoration, the mountain parks across the eastern hills, and the wetland reserves, which those are 19 and 11 are declared as Ramsar places, so they are very important for conservation, and they are inside the city. So the results we got 24 in the biodiversity topic, 12 in ecosystem services, and 45 very good in making laws, Colombia. So this was our best score. And we got a total of 81, as I showed you before. So the main conclusions, we have a great biodiversity in vascular plant and arthropods, but a challenge remains in ecological connectivity. We have a matrix not connected, full of green dots, and we have been neglecting the monitoring of endemic species. Regarding ecosystem services, we have weaknesses in water and climate regulations, since we have lots of not the area that where the water is not absorbed by the earth. I forgot the word. And regarding governance, we have gaps detecting economic valuation of biodiversity. We need more economists doing this. And we, as everyone in biodiversity, we face challenges in budget allocation. And that's it. Thank you. Well, well, in our program of monitoring biodiversity, we propose to measure it every 3 years. So the next update is going to happen next year. So we think we propose this time because it's the time we have to cover the repetition of the measuring of biodiversity across the area, and to see if the application of the policies and programs are working. 3 years is a good time. And what else? Mainly because of that. And the indicator proposed that, yes. So we inferred them, but yeah, I have the calculations here, just because we are a huge team. We have ornithologists, herpetologists, mastozoologists, geese experts who help us with this. But yes, it was inferred mainly, but based on real mapping data. So thank you.
restoration monitoring towards achieving the target 2 of the Kumi-Montreal Global Biodiversity Framework. I'm going to provide a brief background, which was in March 2019, the UN General Assembly proclaimed 2021 to 2030 as the UN Decade on Ecosystem Restoration. And in achieving this, they established 5 task forces, 2 of which are led by FAO. The Task Force on Best Practices and the Task Force on Monitoring. And in parallel, they work with the KMGBF Target 2, which is to restore 30% of all degraded ecosystem by 2030. So I work under the Task Force on Monitoring, and I'll be presenting our work. First, the Task Force on Monitoring has this objective, which is to develop a framework that shows global progress of restoration for the UN Decade and also for the Kumi-Montreal Global Biodiversity Framework. So we already have this in place to report on area under restoration. Also to serve as focal point for providing technical guidance and assistance on restoration monitoring, including to foster collaborations between conventions and frameworks, emerging monitoring initiatives. This is because we also know that there are monitoring platforms out there who are already collecting restoration data. And to reduce reporting burden for countries, we try to work with them through interoperability to be able to show global progress of restoration. Lastly, to identify key gaps on restoration monitoring across all ecosystem types. So currently we have more than 500 members from more than 100 organizations, and everyone are welcome to join the task force on monitoring. So towards achieving the goal of the target 2 and the UN Decade Progress, we currently divide the task force on monitoring into working groups, and this working group are to accelerate actions towards the achievement of target 2. For example, the first working group is the indicator development and monitoring guidance by ecosystem types. Within this working group, we have the terrestrial working group, the freshwater working group, and the marine and coastal working group. All of this working group are working towards addressing the gaps in the different ecosystem types and providing monitoring guidance, including indicators that capture restoration progress. We also have the data and interoperability working group, whereby we work with other restoration monitoring platforms to capture global restoration progress overview and also the data sharing policy, among others. Then we have the third working group, which is the national regional capacity development, whereby we work with government to pilot some of our resources and to really test the framework we have developed at the ground level. And the last working group is the indigenous people restoration monitoring, whereby we work with indigenous people and foster dialogues, and also to equip them with the capacity development needed to monitor their restoration sites. So within this working group, there are resources that backed up the development of the framework for ecosystem restoration monitoring, which are called the building blocks. So within this firm, we have 4 components. We have the registry, we have the geospatial tools, we have the search engine that shares initiative and good practice that's been published online, and we have a forthcoming dashboard. So the dashboard is what we provide the analytic of the compiled restoration data from multiple sources. So it's going to show the restoration progress and also by sources. So we have all of this in place already. And like I mentioned already, we have resources that have been published and serve as building block for firm. So I listed some here, for example, the enabling consistent reporting and monitoring for freshwater, which was just recently launched, that shows how restoration monitoring should looks like in freshwater. So we have other resources as well. And all of these resources are developed in parallel with other ecosystems, such as the terrestrial and the marine and coastal. Also, for firm building block, we have other resources we also leverage on, such as the Society for Ecological Restoration or Continuum of Restoration that provide definition across ecological restoration and rehabilitation. So we actually have fields within firm that enable users to specify if the restoration is ecological or rehabilitation. So we leverage on these resources to develop firm. So we also leverage on the global ecosystem typology developed by the IUCN for users to be able to select the ecosystem type under restoration and provide quantitative progress on the area under restoration, which is the disaggregation. We also leverage on the Aurora indicator to be able to capture restoration progress in terms of their impact across biodiversity, water, energy, and climate. So all of these serve as the building block for firm. So among the firm component, the 4 components I mentioned, the registry, the visualize, the search engine, and the dashboard. This is the registry, and this is how the registry looks like. So within the registry, users get to document their restoration data and good practices. So we have it in 3 sections: the planning and assessment, the implementation, monitoring, and reporting. For the planning and assessment, they document general information. Area and ecosystem, they select the IUCN ecosystem type. They are able to document the area under restoration, the commitment, and disaggregate the ecosystem type under restoration, including providing information on indigenous people territory under restoration as well. Then we have the characteristic tabs whereby we partner with EarthMap for users to be able to run spatial analysis to understand their restoration sites better. Then under implementation, users get to select the activities under restoration, either biophysical or enabling activities. Then we have the monitoring and reporting, whereby users select the indicators that are relevant to the restoration sites, and the monitoring shows continuous annual progress of what restoration looks like in the particular restoration site. So that's the firm registry. And just to provide a brief of firm impact. So we actually show progress on the UN Decade flagship, which is the flagship under the UN Decade on ecosystem restoration. So currently, the firm has been able to capture the total commitment, which is 157 million hectares for 27 flagships, and also show the progress under restoration, which is 32 million hectares. So we have been able to capture this data and also collect geospatial files of the area under restoration, including the impacts of restoration from biodiversity and climate. And within the registry, we also have initiative of over 400 plus and over 1,700 good practices. So this is just a few of some of the impacts we have at the moment. And to talk about capacity building and engagement, so we have been working with country for our pilots test exercise, which is also within the working group. For example, in Latin America, we work with Brazil and Peru. In Africa, we work with Kenya. In Asia Pacific, we work with Philippines. We work with countries, the ministries, bringing different ministries together to pilot the resources we currently have, to work with them on their MBSAP, and to really make sure like we are able to test the framework we have developed as well at the ground level. So aside the pilot countries, we also work with regional initiative, global partners and framework, and the flagship, like I mentioned earlier. For example, we work with Ramsar, we work with Protected Planets. On the flagship, we work with St. Lucia. We also work with AFR 100 to really see how we can work with interoperability and also testing of some of the resources we currently have. So in terms of the impact, we already developed the firm and also developed national strategies for restoration monitoring. We already developed guidance and resources and tools to advance restoration monitoring, and some of these resources are still under development. And also advancing interoperability across framework, which is still under development. And so we are working and continuously working around this. So the lessons learned so far, I would say like ecosystem specific approaches are essential. An approach that really matters to terrestrial ecosystem might not really matters to freshwater ecosystem. For example, the Aurora indicator we use to capture restoration impact is too generic. It captures local metrics, but it's not it's not really unique to all ecosystem type. Also, there needs to be a stronger inclusion of indigenous people for high success rates. There's a need for standardized indicators and data collection protocols, and also transparency and accessibility to data. Then the next step, we are currently working on finalizing the development of the dashboard to show global restoration progress, and we'll be working on interoperability here a lot. And also the Target 2 roadmap event to assess our progress and the next phase of our activities. So we want to host this in 2026 in parallel with the UN Decade event, and also to continue to provide country support and capacity building and continuous development of guidance tools for monitoring. So these are our partners and team and collaborators, and I would like to say thanks to everyone. Thank you for your support. Thank you. Bye.
abiertos son la base para realizar múltiples estudios biogeográficos. Estos datos espaciales abiertos pueden complementarse con información filogenética y ello nos permite establecer relaciones evolutivas entre unidades geográficas. Esto lo podemos llevar a cabo y lo podemos implementar en un esquema de regionalización biogeográfica, y aquí yo les traigo un ejemplo con la región neártica. Esta región neártica es un área geográfica identificada en los trabajos de Sclater y Wallace en 1858 y 1876 respectivamente. Sclater utilizó patrones de distribución de aves para identificar el neártico, mientras que Wallace utilizó patrones de distribución de un conjunto de vertebrados. Hasta el 2021, pues no existía como tal un consenso acerca de cómo debería ser la regionalización para esta parte del mundo. Tampoco existía un consenso acerca de los nombres de las diferentes unidades que conforman el neártico, y pues también no existía un mapa que nos permitiera trabajar directamente con los sistemas de información geográfica. Entonces, es por esto que Escalante y colaboradores en 2021, ellos llevaron a cabo una propuesta de regionalización en la que justamente mencionan que la región neártica se subdivide en tres subregiones. La primera de ellas es la subregión ártica, representada en colores azul, la subregión del oeste, representada en colores cafés y amarillos, y la subregión aleganiana, que está representada en tonos de morado. Ellos también encuentran la zona de transición mexicana, que son estos sitios que son neárticos y neotropicales a la vez. Ellos mencionan que el neártico está compuesto en su totalidad por 29 provincias biogeográficas, y también mencionan el neártico, el neotrópico norteamericano, que está representado en las tierras bajas de México y de Centroamérica. Cabe destacar que ellos tomaron como base dos artículos principales, uno publicado en 1943 y otro en 2017. Aún existe incertidumbre acerca de cuáles son las relaciones evolutivas entre estas provincias biogeográficas, así como de la monofilia de todas ellas. Entonces, por ello surge la necesidad de realizar análisis biogeográficos cladísticos, apoyándonos de plantas y de mamíferos. Básicamente, para un poquito en contraste, este análisis biogeográfico cladístico nos permitirá contrastar este esquema de regionalización biogeográfica propuesto. Entonces, lo que hace la biogeografía cladística es básicamente tomar la filogenia de distintos taxones para reconstruir la historia evolutiva de las áreas geográficas en las que se distribuyen los taxones de interés. Entonces, comienza identificando muchos cladogramas. Esos cladogramas se transforman en areagramas, reemplazando los terminales por las áreas geográficas ocupadas, y bueno, se comparan todos estos areagramas. Toda su información se junta en un areagrama general que nos permite ver cómo está representada la historia evolutiva de esas áreas geográficas. Así entonces, para hacer este análisis biogeográfico cladístico que nos permita representar o contrastar más bien la región neártica, pues en primera instancia consultamos la página de datos abiertos de GBIF, identificando nombres de especies de géneros principalmente norteamericanos. Una vez hecho esto, hicimos unas búsquedas exhaustivas en la literatura para seleccionar pues las filogenias. Seleccionamos 11 de ellas, 5 corresponden a mamíferos y el resto corresponden a plantas. Todas estas filogenias tienen el mayor número de especies monofiléticas norteamericanas, y también consideramos que tuvieran que tuviera una fecha de publicación reciente. Entonces, los géneros de mamíferos que utilizamos fue el género Lepus, Filogale, Microtus, Silvilagus y Vulpes. Como pueden ver en los círculos, en los óvalos, está representado el grupo monofilético dentro de la filogenia. En el caso de las plantas, en este caso angiospermas, nosotros trabajamos con dos géneros, Cypripedium y Fraxinus. En el primer género encontramos 3 grupos monofiléticos norteamericanos dentro de esta misma filogenia. Para el siguiente género, en esta filogenia encontramos 2 grupos monofiléticos norteamericanos. Para el caso de las gimnospermas, en el caso de el género Abies y Juniperus, encontramos en cada una de estas dos filogenias 2 grupos monofiléticos de interés. Para el género Picea encontramos únicamente 1, y finalmente para Pinus encontramos en esta filogenia 3 grupos monofiléticos. O sea, resumiendo, 11 filogenias, de las 11 filogenias obtuvimos 18 grupos monofiléticos. Ahora necesitamos ver o recolectar los datos geográficos de las especies involucradas y para ello, en primera instancia, primero descargamos modelos de distribución geográfica potencial de estas especies de este portal de geoinformación de la CONABIO, que es una fuente de datos muy importante para México. En el caso de no encontrar dicho modelo, procedimos a descargar áreas de distribución geográfica. Para mamíferos nos guiamos de esta lista roja de especies amenazadas de la IUCN, y para plantas, utilizamos esta este portal de datos abiertos, Databasing. Para las especies de plantas que no fue posible encontrar tanto su modelo como su área de distribución, nos permitimos descargar su conjunto de localidades. Todas estas localidades fueron verificadas con diferentes portales de datos abiertos, tal es el caso de eFloras. Bueno, ahora sí, ya para hacer el análisis biogeográfico cladístico de los 18 grupos monofiléticos, construimos los areagramas, como les comentaba, reemplazando el nombre de los terminales por las provincias biogeográficas que les presenté en el esquema de regionalización biogeográfica, pues obtuvimos los areagramas. Y para comparar todos estos areagramas entre sí y encontrar un patrón común, utilizamos esta técnica que se conoce como análisis de parsimonia de grupos primario, en donde construimos una matriz de 229 caracteres por 31 unidades. 29 de ellas corresponden al neártico, una de ellas al neotrópico norteamericano y utilizamos un área externa para enraizar el cladograma. Entonces vamos a clasificar estas 31 unidades con base en estos 29 caracteres que obtuvimos de los areagramas están presentes acá. Entonces, esta matriz fue procesada en TNT utilizando el algoritmo de nueva tecnología de búsqueda y cambiamos la semilla aleatoria 50 veces. Como resultados podemos ver que los 50 areagramas presentaron la misma topología y los mismos valores estadísticos. Entonces, como podemos ver, pues ninguna de las subregiones y la zona de transición mexicana pues conformaron un grupo monofilético. Asimismo, la región neártica tampoco pude evidenciar que tuviera monofilia, ya que Tamaulipas y Chihuahua, pues que se han considerado neárticas, pues están más relacionadas con el neotrópico que con otras regiones, perdón, que con otras provincias biogeográficas neárticas. Entonces, aquí tenemos el mismo areagrama, pero trasladado a la geografía, y con base en estas relaciones evolutivas entre estas provincias podemos proponer un nuevo esquema de regionalización biogeográfica. Sin embargo, como es un trabajo pionero, pues aún hace falta realizar muchas más investigaciones, desde reevaluar los caracteres, incorporar más taxones o incluso trabajar con todas las provincias biogeográficas neotropicales. Sería interesante realizar este análisis o incluso utilizar otras metodologías biogeográficas cladísticas. Y finalmente, como conclusiones, los datos espaciales abiertos, así como los datos filogenéticos analizados mediante biogeografía cladística, nos permitió contrastar este esquema de regionalización biogeográfica. ¿Cómo lo conseguimos? Pues que no identificamos grupos monofiléticos dentro de la región neártica. También es interesante observar que la mayoría de los caracteres no se ajustó a la topología del areagrama general, lo que indica una gran cantidad de homoplasia que en biogeografía representa eventos de dispersión o extinción, entre otros. Y por lo tanto, el 34% de los caracteres sí se ajustó a esta congruencia en que la Tierra y la vida, pues la historia entre estas dos es una sola. Y el 71% de esa sinapomorfía se conservó bastante bien y dicho areagrama debido a la alta cantidad de homoplasia, pues puede no estar representando adecuadamente estas relaciones evolutivas que yo les propongo. Sin embargo, hay que realizar, como les digo, más estudios y pues esto nada más es un primer paso para acercarnos a lo que podría ser las relaciones evolutivas entre las provincias biogeográficas neárticas, así como de toda su monofilia. Estas son algunas referencias. Agradezco a todas las personas involucradas y a todas las instituciones que permitieron el financiamiento. Muchas gracias.
if you go up, it's now. That means
But so everyone like, you know, okay.
Sí, por favor.
Me causa curiosidad saber qué razones cree que hay sobre esa falta de congruencia entre los grupos que escogió con las regiones y por qué cree que el aerograma que escogió no representa del todo bien las regiones neárticas. Estudio.
Sí, gracias por la pregunta. Primero que nada, pues puede ser debido al mismo método que se utilizó, que está sesgado a justamente a ser muy sensible a estos eventos independientes de homoplasia. Entonces, pues eso puede estar generando, a lo mejor que no encontremos una historia compartida. O también puede ser incluso de la misma naturaleza, que en la dispersión, pues realmente pues, nos hemos dado cuenta que realmente es un proceso que de verdad pues, es más importante de lo que nos imaginábamos. Y realmente pues hay más dispersión que vicarianza. Entonces, también puede ser que también mis caracteres que yo utilicé para la construcción del areograma, puede que pues tengamos que tengan que reevaluarse o bien podamos combinarlos con otros o cambiar de grupos taxonómicos. A ti.
I have a question for Noah. Okay. So first of all, I actually have like one sort of comment or like thoughts and then a question. So first, my thoughts is like because your model also includes clustering, that perhaps maybe the clustering caused by dispersal stuff. So it's like sort of mapping the real. It's more of a real occurrence probability instead of a habitat suitability, I would say.
Hopefully a statistical way of getting probability for your larger.
Yeah. Well, like so my thought is like I actually think habitat suitability also like sort of complements the results you have because like for many conservation practices we do need sort of the real habitat suitability because like then you will have then you'll know like what environment you should preserve and what environment like needs like improvement. So I think like sort of like complement stuff, like not actually contradictory, right?
Oh yeah, yeah, for sure. We can talk later.
You probably explained it, but I may have missed it. That pseudo absence factor that you consider in your calculations, is that coming from the presence absence data of the parks? Or is, okay, so you define like probability of absence when you go to the entire region, or how is it works? I may have missed it.
It works just by once you make the whole, correct the SPM translated to parts. If you match it, you can match the predictions in the parts. Should reflect the overall prevalence based on that. You have to do the correctness for pseudo observation, also scale it.